Identification of unknown bacteria in microbiology lab involves multiple steps and tests to narrow down the possible bacterial species. The order that I would follow is first I would do a gram stain. It's a good initial test. It'll allow me to know whether my unknown is gram positive, it looks purple under a microscope, or gram negative, it's reddish pink under a microscope. This will also allow me to determine the shape, the morphology of my bacteria. So is it rod, is it coccus, is it spiral? After that, I would culture my bacteria, meaning grow it on agar. I would look to see if my bacteria produces a certain pigment. Does it like a certain temperature? I would grow it at different temperatures. I would also look for different oxygen requirements. So is it aerobic, anaerobic, facultative? And then finally, the last thing I would do is biochemical tests, metabolic tests. There's so many different tests, so you'd have to narrow it down. But a good test is catalase test. It can differentiate between Staphylococcus and Streptococcus bacteria. You can do an oxidase test to differentiate gram-negative bacteria, and there's so many more.